Hello, everyone. I want to thank Elias Stambler and the organizers for having me. Um, I will talk about my favorite topic, cytosine methylation. As many of you know, cytosine methylation plays a crucial role in development, but also in aging. There are many, many aging clocks, immune aging clocks, proteomics clocks, lipidomics clocks, and so on, metabolomics clocks. I want to briefly remind everyone of the strength of methylation. First of all, there's the intuitive appeal. It is a beautiful finding that the DNA molecule that carries the genetic information also encodes time. It's just a nice finding. DNA methylation really reminds us of the universality of innate aging processes. Why can we make this claim? Well, we have multi-species epigenetic clocks, so-called pan-mammalian clocks that measure aging in all mammalian species. The third intuitive appeal is that these are not just aging clocks, but also developmental clocks, or a better term is life course clocks. You can have one regression model, one clock to measure aging in prenatal samples and in centenarians at the same time. So these methylation clocks truly link development and aging. And then there's the technical appeal of methylation clocks. These clocks are already being used in phase 1 human clinical trials, so they are ready for clinical trial applications. The same clocks can be used for in vitro studies, cells growing in a dish. And as I mentioned, we have clocks that apply to all mammalian species. But the most important question at this point is, can epigenetic clocks serve as surrogate endpoints of human clinical trials? Can you use them as a readout in a clinical trial that lasts, for example, a year or 2 years? And the answer is we don't know yet. It's actually an exceedingly difficult question to answer. But there have been some developments. And let me rephrase things. A much easier question is, test whether interventions that slow down the rate of aging, emphasis on rate of aging, whether those are associated with longer health span and lifespan in humans. And when it comes to calculating a rate of aging, you really need longitudinal studies. And I want to draw your attention to a recent preprint by Perry Kuo and Luigi Ferrucci, who looked at almost 700 participants of the Inchianti cohort, an Italian cohort, where they had 2 or more blood samples collected several years apart, and they could calculate rate of change in epigenetic clocks. And here on the right panel, you see several epigenetic clocks, Grimm age, pheno age, so-called second generation clocks. And the hazard ratio that I report here is associated with rate of change, the increase in Grimm age, for example. And you see it is predictive of shorter human lifespan. So these are Cox regression models of time to death. So I've seen now this study and a couple more that make me hopeful that yes, these second generation clocks capture causal association with aging. I want to shift gears and talk about a totally different topic, quality of marital relations. And this study is a study from 2014. I was not involved. And it has nothing to do with methylation. It's just a meta-analysis of whether happy marital quality is associated with better health. And the authors conclude that yes, there's an association. You see here association with cardiovascular related mortality. And so, yes, if you are lucky enough to be in a happy relationship, chances are this has benefits. However, the effect sizes were exceedingly small. And my conclusion from reading the paper is that it's better to be in a bad relationship but have a healthy lifestyle than to be in a good relationship but follow a bad lifestyle. So that's what I want to tell you. But I was very interested in studying the effect of relationships on epigenetic age. But I chose a species where you can arguably control confounders better. So there's an interesting rodent called the prairie vole, and this rodent forms monogamous relationships. And now you can study whether voles or these rodents that are in a happy relationship, they found their partner. You can compare those to voles that never found a partner, so-called sex-naive animals. And in this collaboration with Alex Ophir, we looked at many different tissues, blood, brain, ear, liver, and large sample sizes. And basically, we did not find any evidence that the pair-bonded animals differed from the sex-naive animals when it comes to epigenetic clocks. So just no effect. Moving to another topic, just to remind everyone, there is an intervention that rejuvenates epigenetic clocks in a drastic manner, and this is epigenetic reprogramming based on the Yamanaka factors. This finding has been known since 2013. iPS cells are perfectly young, but we also find it with our universal pan-mammalian clocks. Now, the question is whether interrupted reprogramming could also have a rejuvenating effect. And here I show you results from a recent study by Rudy Goya, where he injected an adenovirus with OSKM into the hippocampus of rats and observed then a benefit. Memory performance was improved in a Barnes test, but also we found evidence that the hippocampus was rejuvenated according to several epigenetic clocks. So for me, it's quite interesting that this Yamanaka therapy also applies to brain regions. Oops. I want to talk about so-called third generation epigenetic clocks. So what are those? These are multivariate regression models that predict relative age of a species, where the relative age is defined as the ratio of the age of the animal divided by its maximum lifespan. And this mathematical operation leads to a value between 0 and 1. For example, the relative age of a 61-year-old person is 61 divided by 122, so it's 0.5. And next you can form these relative ages of different species and build a predictive model based on highly conserved cytosines. And this actually works. So here, I want to show you a recent study where we build clocks that apply to amphibians, axolotl, and frogs, the so-called xenopus, the clawed frog, and humans at the same time. So in the panels, I show you on the x-axis the relative age of the species, And on the y-axis, I show you a leave one out estimate based on methylation. So it's, for me, remarkable that you can build these methylation clocks that apply to these very different species. And it's particularly, I like that finding because it shows that there are certain aspects of aging that are truly conserved in vertebrates. Now, there was a curious finding for the axolotl, which was the following, that, so yes, we were able to build epigenetic clocks for axolotl, but only in animals that were younger than 4 years old. By contrast, animals that were older than 4 did not allow us to build an epigenetic clock. And that's a very curious finding, but it supports the hypothesis or the idea that maybe the aging process is halted in these axolotl. So yes, development happens, but then epigenetic aging stops. So this finding may or may not relate to the finding that the axolotl has this remarkable ability to regenerate tails and limbs and other organs. So this is work with my collaborator Max Yun and Julia Halusa. Now I want to talk about maximum lifespan. Famously, the oldest human was a smoker. And so if you just studied maximum lifespan in humans, you would perhaps jump to the conclusion that smoking is actually good for you. But that's not the case at all. But I show you this finding because one of the punchlines of my research is that whatever biological processes relate to maximum lifespan, these processes are quite different from those that relate to individual mortality risk or average lifespan in humans. And the reason is maximum lifespan is a species characteristic, whereas our mortality risk really depends on the individual. So I started the so-called mammalian methylation consortium that involved 200 collaborators, and we generated and released data from 348 mammalian species, 15,000 methylation array samples, all publicly available. And here is the long list of collaborators from all across the world. And we carried out epigenome-wide studies of maximum lifespan. And one of the interesting things was that the lifespan-related cytosines were enriched with binding sites for OCT4, SOX2, and NANOX. So these pluripotency factors for stem cells. So to me, it's remarkable that this study of maximum lifespan relates to these pioneer factors that relate to reprogramming and pluripotency. We have a very good understanding of the methylome in long-lived species. So long-lived species are very good at maintaining a mountainous profile of methylation. Low methylation in transcriptional start sites and bivalent promoter regions, but high methylation in gene body and intergenic regions. By contrast, short-lived species have a flat methylation landscape. We developed a multivariate predictor of maximum mammalian lifespan based on cytosine methylation. And this predictor of maximum lifespan is, again, remarkably accurate on the log scale. So if here I show you log transformed maximum lifespan versus the predicted maximum lifespan. And using this biomarker, one can test, well, which interventions seem to affect maximum lifespan in the mouse. And there was a hint for dwarf mouse status. So you may know there's certain knockout mice that are dwarfs, and they live a year longer. And so we saw a hint that this could affect maximum lifespan according to our methylation predictor. But there's another famous intervention in mice, caloric restriction. And in 4 out of 5 data sets, we saw no effect. And it's well known that caloric restriction relates to mortality risk in mice in about 1/3 of mouse strains. Only 1/3, but not in all. But anyways, it does not seem to affect our prediction of maximum lifespan. And that again makes the case that whatever relates to the species characteristic maximum lifespan is quite different from what relates to the individual mortality risk of a mouse. And when we looked at cytosines that relate to maximum lifespan, they really had very little overlap with cytosines that relate to chronologic age in mice. So quite different. And I was frustrated by the fact that aging effects did not overlap with maximum lifespan. And so we recently published a paper where we link maximum lifespan to aging effects in specific chromatin regions. And this leads to the important technical part. We needed to define rate of change of methylation. So again, rate of change with age. And the hypothesis is that species where you have a fast rate of change in methylation, those may have a shorter maximum lifespan. And this hypothesis is true as long as you look at the right chromatin states. So we use the so-called chromatin states based on histone modifications. And so yes, we do find that rate of change in methylation relates to maximum lifespan, but only when you look at specific regions repressed, for example, by the polycomb repressive complex 2, so bivalent regulatory regions. And here I show you how log lifespan relates to rate of change in methylation. I think I'm out of time. I want to stop. And I started a non-profit foundation that can generate mammalian methylation data for anybody. And I'll stop with that. Thank you very much.
Thank you very much. Are there any questions from the audience?
No questions, only compliments. You make a great job, and it could be said that your clocks could be a basis for quantitative evaluation of rejuvenative events, rejuvenative interventions, and so on. So good luck, and thank you very much.
Thank you very much.
I echo with that, of course. Thank you very much for being the pioneer of longevity, and we all use your clocks, so thank you.
There's a question in the chat by Miriam O'Reilly.
Yes.
Can you provide information on the non-profit, maybe a link? Just do a Google search for Epigenetic Clock Development Foundation in Torrance, Los Angeles. The CEO is Bobby Brook. It's a non-profit, and anybody can get this mammalian methylation array, or simply email me. Yeah. But thank you all, and next time I hope to meet you in person. Bye-bye.
Hopefully. Bye-bye.